Hi everyone, my name is Mia, and in this video I'm going to show you how to analyze codon usage and nucleotide composition using the hexokinase A gene from Drosophila melanogaster, and I'm going to show you step by step how to compute every Excel formula and also the interpretation from a biological perspective. This is the dataset that we will work with. The full sequence has 4,139 nucleotides, so let's analyze it. We're going to start with a frequency table for each nucleotide for adenine, thymine, guanine, and cytosine. We're going to use this table over here, and we're going to use the formula count if, where we consider the range and the criteria. We select our entire sequence, we know which is the last cell of our sequence, so we just type it in, and we select the first nucleotide, adenine. Now we're going to lock in the range of our sequence to make things easier, because we're going to use the same formula for every nucleotide, just changing the criteria for each nucleotide. And when we pull down our value, we will have the frequency for each nucleotide. And when we add them up, we will see if we consider all of our sequence. And you can see we obtain our desired value. The relative frequency basically will give us the percentage of each nucleotide in our sequence, and we obtain it by dividing the frequency over the total amount of nucleotides. And we do the same here, we lock in the total value. So we just pull down our first number and get the relative frequency of each one. We have to sum it all and get 1 to see that we consider everything, and we convert our values to percentage. Is each nucleotide equally likely? No, clearly not. So we're going to address this question in more detail using hypothesis testing. To do this, we will test two hypotheses. Our null hypothesis will be that each nucleotide is equally likely, 25% each, and our alternative hypothesis that they are not equally likely. Okay, so our expected value, meaning the frequency we will expect if each nucleotide was 25%, would be our total amount of nucleotides over 4, the 4 nucleotides we have. To obtain our chi-square value, we also need the degrees of freedom. We consider our 4 categories minus 1, so we obtain 3 degrees of freedom. Now that we have everything, we will need this formula over here, where basically we will subtract from the frequency of the nucleotide our expected value, then square that value. I use the function power here. And finally, we divide everything again with the expected value. And as you saw in the formula, we need the sum of each value of each nucleotide in this case. So we log in our expected value like this. And when we pull down, we will obtain all the values we need. Then we sum them and we obtain our chi-square value. And to obtain our p-value, probability, we'll use this formula where we consider x, the sum of our chi-square, and the degrees of freedom, that in this case is the number 3. And as you can see, the number we obtain is too small, that's basically 0. Here I'm going to add some decimals for you to see that there's a number, but really small. So a chi-square value that is large and a p-value very small, less than 0.05, mean the difference between nucleotides are not due to chance. The distribution is biased. So we reject the null hypothesis of equal nucleotide distribution, and we can conclude that the sequence has a real nucleotide imbalance. Now we're going to calculate the CG content in our sequence by adding the frequencies of both the guanine and cytosine and dividing it by the total amount of nucleotides we have. We have almost 45% of CG content in our sequence. So we have a moderately CG-rich content, And in Drosophila, this content is biologically meaningful because a higher GC content correlates with a preference for specific codons, which are the favored codons for many highly expressed genes in this species. Now, talking about codons, how many codons does our sequence have? Well, we have 1,379, and we obtain this by dividing the total amount of nucleotides over 3, because each codon has 3 nucleotides. But to separate our sequence in codons, it's way easier to put our entire sequence that is in one column in just one cell. So we're gonna use this function. So our delimiter will be empty, so we put quotation marks because we don't want any spaces or commas between our nucleotides. Then we will put true because this tells Excel to ignore empty cells, and then we select the range, our entire sequence. And once we have our full sequence stored in one single cell, we need to split it into codons in groups of 3 bases. So to get the first codon, we will use this formula. So we type it in one cell, and first we select the cell where our sequence is. Then we put 1 because we're going to start at the first nucleotide, and the length will be 3. So as you can see, we have ATG, adenine, thymine, and guanine. Now we're going to use this formula over here to get the other codons. Here we use the expression row because we want to create a counter that starts at 0 in the first row and then becomes 1 in the next row, then 2, and so on as you move down. So this counter is multiplied by 3 because each codon is 3 bases long, which tells Excel how many characters to skip. And finally, adding 1 adjusts the starting position because myth begins counting at 1, not 0. And as a result, the formula automatically will extract characters in the first row, then starting the fourth one for the second one, and so on to get the full sequence. So here we pull down the value we got here. So as you can see, we will get the entire sequence divided in codons. We have to pull it down until we get nothing. So we finish with all of codons.
2 hours later.
We finally get to the end of our codons, and we have a leftover of 2 nucleotides that cannot form a codon, of course, but here it is. Finally, we will elaborate a codon frequency table. You can find this list of codons and the amino acid they code for in the internet for the 64 codons, and simply paste it in Excel. So we will use the formula COUNTIF again for each of the codons. Okay, remember you have to select the range, the entire codon list, and when you lock in the values of the range, you can do again what we did before, that is pull down the value, so it will automatically change the codon you are looking for. And finally, remember we will add them up to see if we have the amount of codons that we expect with our calculation before. So we select all the cells with the frequencies of codons, and we can see we obtain exactly the number we wanted. I made this table just to analyze and compare synonymous codons, and for each amino acid I found the most common codon that code for them and the total amino acids coded for each one. So we can observe some things here. The ones that are bolded are the most common ones. Specifically, the arginine and leucine are the most common amino acids found in this gene. Now, it is important to remember that the hexokinase gene has an important role in glycolysis. So these amino acids that are frequent, they're not frequent just because they have an important correlation with the functionality of the enzyme and the functional needs it has. So positive residues like arginine and lysine help bind ATP with phosphate, and other hydrophobic amino acids like the leucines give this stability to the protein, and other small residues that, for example, alanine or glycine, that are also frequent, provide this flexibility required for catalysis. So overall, we can observe a clear codon usage bias. Synonymous codons are under selection. We can observe the GC content that shapes these codons that are preferred over others. So this is really important, talking about what the gene function is and what proteins they code for. So that's it for this video. I hope you really enjoy it and could understand not just the formulas and computations in Excel, but also the importance and significance of this kind of analysis and data interpretation in science. So if you have any questions, don't hesitate to write it in the comments, and thank you for watching.